Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Mazida Nurmania Rizki and you can call me Zida. In here I want to present a biochemical report of the UREASE test. So watch this video carefully, okay? Through this video I will convey the point of the research I did. The first is the introduction, second is the background, third is the method, fourth is the result and discussion, and the last one is the conclusion. The first is the introduction. Biochemistry is the study of chemical processes that occur in plant, animal, and microorganisms, as well as the change they undergo during development and life. This biochemistry was first introduced in 1911 by Carl Neuberg, who was a chemist from Germany. One of the exciting studies in biochemistry is about enzyme. Enzymes are a group of proteins that carry out and regulate chemical change in biological system. One of the functions of enzyme is as a biocatalyst. Enzyme work can also be inhibited by several things, including excessive heating, which will damage the enzyme. The addition of inhibition in the form of heavy metal will also damage the enzyme so that the enzyme will experience denaturation. Okay, let's get into the discussion about the background of the research. One of the enzymes found in plant is the urease enzyme. This enzyme will be able to break down urea into alkaline ammonia and will produce a product in the form of carbon dioxide. There are several indicators that can detect the presence of base in sample. One of them is through the use of phenolphthalein, which is an alkaline indicator. The addition of this indicator can cause the solution to turn pink color. And then the experiment, the phenolphthalein indicator will prove whether the urease enzyme work well in breaking down urea or not. Later, different treatment will be carried out on each tube, such as the addition of heavy metal inhibitor and heating to boiling. Okay, let's get into the research method. The research method, the method used in this research is a laboratory experiment which data collection through analysis of the result of the practicum. The research was carried out in the Biochemistry laboratory on May 12, 2022 at 1 p.m. and the material in this study you can see on the screen. They are about dispersion solution and other. So we're gonna next to slide. There are about the fourth point in the study is the result and discussion. You can see the result of the experiment tabular in the tabular form on the screen. And then you can see through the documentation of the result of the urease test experiment. There are the point about the result and this is about discussion. And tube 1 is show a pink color. This is because the ammonia compound produced from the breakdown of urea by the urease enzyme is alkaline, so the pH will rise, and as a result the phenolphthalein, which was colors turn pink, which in this case can be said that the enzyme is working normally. So it is proven that urea is broken down by the enzyme urease into ammonia and carbon dioxide. In tube 2, the solution will be heat on so that the enzyme will be damaged and denatured. Discuss the urease enzyme cannot break down urea into ammonia. In the tube 3 is so clear and cold color. This happened because the urease enzyme cannot break down urea due to the addition of an inhibitor in the form of HgCl2. So that the result and discussion in this experiment, then we can come to the conclusion. The conclusion of this experiment is that soybean suspension contain urease enzyme because the phenolphthalein indicator show a pink color. The performance of the urease enzyme from soybean suspension can be inhibited by heating at 100 Celsius and by an inhibitor in the form of heavy metals such as Sag2+, Pb2+, which cause the enzyme to denaturate. So that's my presentation. Sorry if there are any mistake. Wassalamualaikum warahmatullahi wabarakatuh. Bye.